A very good morning to everyone. I'm happy to welcome you all back to Agam webinars. And today it's going to be about the anatomy of retina. So we all know that retina is the innermost lining of the eyeball. And we know that the picture that what we are watching, it falls on the retina, and that's what creates the picture, right? The image on the brain. Now, what exactly is happening there in that very thin layer? We need to know that, right? So, guys, how many of you can hear me? How many of you can see the screen? All right. So, if you ever wondered how this retina is functioning, we can correlate this eyeball, the retina, to an artificial device that we all have called camera. So we know how a camera functions. The light that is there around us is collected or gathered by the lens, and this lens will direct the light onto the screen. Okay, so that's what we see as a picture in the screen, and that's how we get a picture. So the same thing happens inside our eye. The lens in the eye is going to collect or gather all the light around us, and that brings the light to focus on the retina. And the retina then processes this to convert it into some impulses, electrical impulses, and that impulse will reach the brain. So this is what is happening in the eye, like a camera. But to add more correlation um to the structure of retina, I would like to relate this with the cinema screen, right? So everybody will love watching movies in the theaters, and so we all know that in theaters we have a huge screen, and on the screen we'll watch the movie. So like in childhood days, we would not know how it's happening. It will be a wonder for us, but in the time being, we would have understood that there's a projector at the back of the theater, and that's what is projecting the light on the screen, and that's how the picture is created, right? So the same thing is happening in the eye. So when you are sitting in a seat in the theater, just correlate with your eyes. In your eyes, you are sitting in the posterior segment of the eye. Okay, just correlate this. When you are inside a theater, it's like you are inside the posterior segment of the eye. We'll all see those structures in a short while, but just imagine. Now, standing in the posterior segment of the eye, you can imagine yourself that, you know, here's the posterior segment of the eye. Okay. Yeah. So most of you might know about the parts and all, those basic parts. Okay. So we know that this is the posterior segment. Okay. This zone is called the posterior segment. Now, in the posterior segment, when you are standing in the posterior segment, you can see that the light from the lens is going to fall on your retina. Okay. So this is same as the light from the projector is going to fall on the screen. Okay. So you are in the theater seat. So this is, you need to imagine it now, right? Then we'll see about what is there in this retinal layer, and what are the structures or some specific parts in this retinal layer. Okay. So before beginning into that anatomy of eye retina, let's have a look at the preview today. Okay. So first we'll see how this retina is having its structure. Then we'll see about the histology of retina. So histology in the sense we'll see about the layers of retina in detail, and then we'll see about rods and cones, which are specialized photoreceptors in the eye, and we'll see a little about the blood supply, then clinical correlation, and finally, as usual, we'll see the important stuffs for your exams. Okay. So guys, how many of you can hear me? Just tell it in the comment so that I can continue the session. Okay, so right now what we are seeing is the cross section of the eyeball. So in the cross section of the eyeball, you know the basic parts. So let us just give a quick recap for those basic parts. So first, the outermost layer is called as the sclera, right? The lining layer inside the sclera is called choroid, and within the choroid we have retina. Okay, so retina is the innermost layer, the very thin, sensitive layer. Okay, so now our focus is going to be on this retina. Now, retina is a screen, right? So the screen is like a lining. Now, this lining has two important layers, right? Functionally, based on the performance or the activities performed by the cells in these layers, we divide them functionally into the outer pigmented layer and the inner neural layer. Okay, so this particular part or this particular yellow zone is the retina, right? I'm just highlighting it here. This zone is the retina. So if you keenly observe, you could have seen from where I started. I started this marking from here. Okay, what point is that? We could say it is the junction between the choroid and the ciliary body, right? Ciliary body is this part. This muscular part is ciliary body, right? And continuation of that in the posterior aspect is the choroid. So in between the choroid and the ciliary body is where the retina actually starts. Okay, I mean the main part of the retina actually starts, right? With this, we'll just zoom in into the layer of the retina, right? So we have got two important layers. The outer pigmented layer, as I mentioned you, this pink color zone is the outer pigmented layer. Okay, now that is thin when compared to the inner neural layer, right? Why is it so? Because of the number of cells, right? So why do we say that way? The number of cells. So yeah, we have got, actually speaking histologically, we have got 10 layers in the eye retina. So there is one pigmented outer pigmented layer, and there are about 9 neural layers. Okay, 9 layers within this inner neural layer. So it is like this 1 plus 9, which constitutes totally 10 layers in the eye retina. Right? So there are totally 10 layers in the retina histologically, but fundamentally you can put all those 10 layers into two groups, the outer pigmented and the inner neural. So from the name we can imply the pigmented layer is on the outer aspect. So it is having some pigmentations in it, the coloring agents in it, right? So it is called the pigmented layer, and within that the neural layer. So it has all those neuronal cells, like the ganglions, the photoreceptors, the other nerve fibers, all those things. Okay, so for now, just remember these two layers of eye. In a short while, we'll see about the histology of eye as well. Okay, so by that time, we'll elaborate about all those 10 layers. Now, let us appreciate one more thing in the eye's retina. So what I said you is layers of retina, right? Now, we can also divide this retina into two different parts, right? Two different parts in the sense. On the posterior aspect, we have an optic part. Optic part means that is a sensitive part. So that is the layer which actually picks up the light and converts it into some electrical impulses. Okay, so that's called as optic part. And anteriorly, the retinal layer becomes even thinner and it forms the non-visual part of the retina. Okay, so let me show that here in the picture. What you're seeing is the yellow color zone is the inner neural layer. Okay, and this blue color is what you are seeing as the lens. Now this inner neural layer is progressing forwards as thinner one. Okay, it gets thinned out. All those 9 layers, it gets decreased in number. It becomes 8, 7, 6, 5, 4, 3, and finally it becomes very, very, very thin, that it doesn't function the conversion of light impulses into electrical impulses, right? So the thing happens here on the top and the bottom. Okay, now what happens to the outer pigmented layer? It is the outer pigmented layer which actually continues on the anterior aspect. So this was the posterior lining. It was very thin anterior aspect, it would even disappear, right? And the pigmented layer, as I'm showing you now, the pigmented layer, as it proceeds forward, it is the thicker one. As it proceeds forward, we know that the ciliary body continues in front as the iris, right? So it lines the iris. What lines the iris? The pigmented layer. And one more thing, we all know that on the outer aspect, what do we have? Outside the retina, we have the choroid, right? So here is a big layer, the thick layer, choroid. Okay, it's lining entirely. Okay. So this choroid is what comes in the front, and then it forms this, it divides into anterior and posterior segments. In the anterior aspect of the lens, we have the iris. So that thinner part of the choroid is the iris. So here's the huge ciliary body. Okay. Now this is what happens. The pigmented layer is going to line this ciliary body, and on the anterior aspect, the iris as well. Okay. So this thing, the pink colored one and the yellow color one, I've just shown you how it gets thinned out and forms the lining over the choroid, and then after the choroid we have ciliary body, and then in the anterior aspect we have the iris. So it's lining all of them, right? A very thin lining. So still it's also a part of retina, but that does not take part in the activity of converting the light impulses into the electrical impulses, right? Okay. Now what is actually separating this optic part and the non-visual part? Okay. So what is that particular zone called as? We call that as ora serrata. Okay. It's like this. It's actually an irregular lining. Okay. It's an irregular margin or a boundary called ora serrata. So we all know serraceous or serrata means irregular, or like it's like knife. Okay. If you've seen in the knife, you would have seen those irregular border, right? So that is called as serrata. So this lining is quite irregular. So we call it ora serrata, right? So I've just marked it in green color. So this green color marking, this ora serrata, is the distinguishing lining between the optic and the non-visual part. So it's like this. Okay. Now where does the ora serrata lie? The ora serrata lies between these two parts. So outside, how can we relate this? It's between the choroid and the ciliary body. Okay. Just know these facts, and that's enough about the parts of retina. Now let us see some specialized regions in the retina. And yeah, one more thing. Now that we have seen only the cross section, I would also like to appreciate one more thing. So if you would take the anterior aspect, see, this is the choroid, and within this we have the retina, right? So within this we have a retina, and the retina gets thinned out in the anterior aspect like this. So how does this ora serrata actually look like? It's a margin, right? So this will look like this. Okay. So the thing is, see, just imagine, look at my hands. This is the eyeball. Imagine this is the eyeball. The outermost layer is the choroid. We are not talking about sclera now, so just leave it right now. So this choroid is lined on the inner aspect by the retina. Now this retina, as it comes to the anterior aspect, at the junction of choroid and the ciliary body, there is an irregular margin or the lining. So that looks like this. Okay, it just forms an margin that is irregular, and that margin we call as ora serrata. After the ora serrata, this retina gets totally thinned out, and then from the anterior aspect, it will line the ciliary body and the iris. Okay, so that's about the lining. Hope you got a good imagination of how this retina is lining. Now that we have talked about the main parts, we'll go to the specialized zones or regions in the retina. Okay. So have a look at these three terms that I've marked here: optic disc, macula lutea, and fovea centralis. Okay. There's no more fancy terms. If you just remember these three terms here, that's enough for the retina. Okay, there's no other fancy terms. So first, let's see where this optic disc, macula lutea, fovea centralis is located. So the retina is forming a lining, right? So the centermost part of the retina is called as the macula lutea. Okay, what do you call it? Macula lutea. I'm highlighting that in green color. Okay, right. So see here, this region, this zone, okay, this zone is what forms the macula lutea, the centermost part of the retina. Okay, and the centermost part of the retina, within that we have a centermost point. Okay, that particular point is called as fovea centralis. I'm marking that in pink color. So this point, okay, this point, it looks like black. So that particular point is called as fovea centralis. So what you need to remember is this: the centermost point of the retinal lining is called as the macula lutea, and the centermost point of the macula lutea is called fovea centralis. Okay. Now, where is the optic disc? So, optic disc is where the nerve leaves the eye, and that is the point where the central retinal artery enters the eye. Right. So, this is the zone of optic disc. Okay. So, this is the zone of optic disc. Okay. What is the distance between this fovea centralis and the optic disc? It is better if we have gross idea about that. It's about 3 to 4 millimeter distance between the fovea centralis and the optic disc. Now, as you hear the numbers, not just here, anywhere, when you learn about some number or such facts, just imagine it with your real-time life, okay? So, let's say we are telling that it is 3 to 4 millimeter, right? So just compare it with that scale, okay? You have a scale where you have just only 3 to 4 millimeter. It's that small. The distance between those two is that small, right? It's quite relatable because the eyeball is very small. So within that we have the retina. Within that we have these two small points. Between them, the distance is 3 to 4 millimeter. Okay, that's enough if you know these points. Okay, and one more thing. The types of vision that we have is color vision and the dark vision. So why we call it color vision and the dark vision? So color vision is the bright light vision. That is that which is there in the daytime, right? And the dark vision is that which we can experience in the nighttime. Because we have these tube lights and artificial lights, we don't experience it. But as we go out where there is no light, our eye has to depend on the minimal light that's available. So we call that dark vision. So this light vision and the dark vision can be appreciated on the retina as well, because certain zones in the retina focus on the bright light, and there are certain zones which do not focus on the bright light, and they mainly focus on the dim light. Okay, so what are those zones? So whenever I speak about the color vision, you just know that we are talking about the RBG. What is RBG? Red, blue, green, the three primary colors. Okay. So the color vision is mainly focused around the macula lutea. Okay, I'm marking here. The macula lutea is the zone where color vision is concentrated. So in your daytime, it is the fovea centralis or the macula lutea where the maximal functioning is there, right? And in the night time, in dim light, it is the zone peripheral to the fovea. Okay, so the peripheral zone is what is functioning more in the dim light. So the black and white vision or the dim light vision is on the peripheral zone, right? And one more thing I'll mention now. The cones are the cells, the photoreceptor cells, which are mainly meant for the bright light or the color vision. So those are concentrated in the fovea centralis. Keep that in mind now. In a short while, we will see them in detail. Okay. So I just mentioned you that the retina is forming a very thin layer on the anterior aspect, and that's called the non-visual part. So we should know how this is actually happening. For that, we'll see a little bit about the embryology, right? So in case you know already about the neural tubes, this picture will be familiar for you. If not, it's not a problem. So have a look at this. This is the structure of a neural tube. Okay, this is a section of the neural tube, the sagittal section. Now, what are we appreciating here? See, this is not at all a complicated thing. The names are quite different, like telencephalon, diencephalon, metin, myelin. Okay, that's not a big deal now. Just think of this thing. You know that brain has three parts: forebrain, midbrain, and the hindbrain, right? So the same parts, when we are as an embryo, we call them by a different name. Okay, that's it. So the forebrain is technically called as prosencephalon. Then we have mesencephalon, which is the midbrain, and we have the hindbrain. Okay, so the hindbrain is called as rhombencephalon. Okay, so now from where does the retina develop from? I'm not going to concentrate on the rest of the eye's embryology. It's going to be only about the retina's embryology. So from where does the retina develop? It develops from a structure called diencephalon. What is diencephalon now? Now, diencephalon is actually a part of prosencephalon. You can see it on the left side. It is marked here. Diencephalon and telencephalon are parts of prosencephalon or forebrain. So in the diencephalon, that is the place from where the retina develops. So let's diencephalon. This pink color lobe is the diencephalon. Now, from this pink color lobe, we are going to see how we are going to see how this retina is developing. So first part that protrudes from the diencephalon is called as the vesicle, the optic vesicle. Okay, right. Okay, so here from the diencephalon, the optic vesicle develops. Okay, it protrudes out. Then what happens? This protrusion, as it progresses, it forms a groove or a depression in the center, right? Just like this. Now, the groove then deepens further to form a cup-like appearance called as the optic cup. Now, the optic vesicle has become the optic cup. So, this protrusion was called as optic vesicle, and now it has become the optic cup. So, in the optic cup, what happens is, as it further progresses, this anterior end will thin out, okay? And only the pigmented layer, we mentioned the outer pigmented layer, right? So, only that layer will come forward and it will form a lining over the ciliary body and the iris. So ciliary body, iris, all of them develop from different structures. We don't need that now. For now, just remember how the retina is developing. It is from the diencephalon. It protrudes as an optic vesicle, which further forms a depression in the middle, right? And the depression or the groove progresses to form the optic cup, and the optic cup slightly grows forward and it forms, it gets thinned out in the anterior aspect. That's what differentiates this optic part and the visual part, right? So we can just mark it here. Guys, do you remember what name did I mention? What is the lining or what is the margin that separates the optic and the non-visual part? Can you remember? So what is the lining that separates the optic part and the non-visual part? What do we call it? Okay, yeah, very good. Priyanka, Salla, SVN, Learning Desk, Harini, right, okay. Very good, guys. So that is ora serrata. If you can remember that word, then it's very easy for you. There's no other difficult word or a confusing term like that. I mean, it's new for you, so it will be a little different. Okay, so that is ora serrata. Right. So this lining, everything develops the time when the optic cup is completely developed. So this is about the embryology of retina. Now let us see about the most important part of this session today. Okay, this is a very important short note for you. Okay. It is the histology of retina, or they will ask you as the layers of retina. What you need to write in that answer is all these 10 layers. Okay. All these 10 layers. Okay, so if you just read these terms just like that, you will not remember, right? So if you understand the meaning of why are these layers arranged this way, then it will be very easy for you, right? You don't even have to revise it before the exams. So just now, listen to me. I'll tell you how these are arranged and why they are arranged that way. Then you will know why we have these many layers and why do we name them in such a manner, right? So yeah, in the beginning I mentioned you there are only two functionally important layers. So we are splitting this 10 layer into 1 plus 9. The one is the outer pigmented layer, and all those 9 layers together form the inner layer. That is the inner neural layer. So neural layer which has all those neural cells like glial cells, ganglions, photoreceptors, so many other cells are also there. So this is the outer pigmented layer. It is very easy because it is just one layer. So have a look at this diagram. This part is the outer pigmented layer. Okay, right. It's very beautiful. Outer pigmented layer. Right. Now we'll focus on the other layers. So if you need to remember them in the order, I'll just tell you about a small mnemonic, right? So first, before getting into those layers, let us know about some special cells in this retina. What are the special cells here in the retina? Remember, in the same order, we have rods and cones. I mentioned you, rods and cones are the photoreceptor cells, right? So photoreceptors, rods and cones. Second, we have bipolar cells. Okay, bipolar cells means they have these synapses on both the ends of the nucleus. So we know a cell has a cyton, a neural cell or a neuron has a cyton and an axon. So you can imagine if the cyton is having two axons on two opposite ends, we call it as a bipolar cell. So there is bipolar cell. Then the third type of cell is called ganglion cells. Okay, and the fourth type of cell is horizontal cells. So as the name implies, it will lie horizontal. Then amacrine cells. Okay, so amacrine cells is the fifth type. Okay, now just remember it this way: RBG, ha ha ha. Okay, so if you remember it that way, you will not forget it at any point. So remember it. It is RBG, ha ha ha. That is HA. Okay, so this is in the order from the outer pigmented layer towards the center. Okay, which means immediately you need to correlate it with the eyeball. So outer pigmented layer is the outer aspect and the inner neural layer is on the inner aspect. So what I'm telling as RBG is from the outer pigmented layer. From there we are getting towards the eyeball. Okay, so it's RBG, ha ha ha. Okay, now let's see where this R is there, B is there, and G. Look at this diagram. Let's mark the RBG. Okay, so this layer, you can very well appreciate the rods and the cones. So this layer, okay, in this layer, we have the rods and cones. Okay, so which is a rod and which is a cone? As the name implies, the rod is a rod-shaped structure. This is the rod-shaped one. And the cone shape, the orange color one is the cone. So these are rods and cones. Then after R, what did I say? There is B, right? So B is the bipolar cell. This red color one is the bipolar cell. Okay, and then below this bipolar cell, G indicates the ganglion cell. So here we have the ganglion cell, right? Okay, the other two cells we mentioned are horizontal cell and the amacrine cell. So here is the horizontal cell. So first you need to know these three things. So even when you draw the diagram, if you keep this in mind, it will be easy for you to draw. First layer, first we are going to draw the rods and cones. Then we have the bipolar cells. Below that we have ganglion cells. So in between rods cones layer and the bipolar cells, we have the horizontal cell. Okay, of that ha ha ha, we have this horizontal cell is H, and the amacrine cell. Where is this located? It is between the bipolar cell and the ganglion cell. Okay, so that is amacrine cell. Okay, so once you have located where all the cells are located, are positioned in this layer, further on we can proceed to other structures. Okay, so now what have we finished out of this 10 layers? we already saw what is outer pigmented layer. Then out of the 9, now we have covered the layer of rods and cones. Then we have seen this ganglion cell layer, right? The innermost cell that we mentioned. And we mentioned about the bipolar cell, right? So we cannot show it here because it is not named based on the cell's name. We'll see that in a short while. Okay. So first, rods and cones. So rods and cones form a plexus. Plexus means the nerve fibers are forming some, they have some interactions at the synaptic cleft. Okay. So when they form such interactions, we call that the plexus. Okay. So based on the term plexus only, we call it as plexiform layer. So where there is a plexus between the rods and cones and the inner nuclear layer, the bipolar cells, we call that the outer plexiform layer or external plexiform layer. Okay. So let us not concentrate on the horizontal cell and amacrine cell now. Leave that now. We'll just talk about the rods, cones, bipolar, and ganglion, only about the RBG. So in between the R and the B, we have the outer plexiform layer. And in between the bipolar cell and the ganglion cell, there is a plexus again. And this plexus is called as inner plexiform layer. Okay. Right. Now, now we have completed 2 layers. What are the 2 layers we've completed now? The outer plexiform layer and the inner plexiform layer. Okay. Now, we need to see the other layers. The nuclear layer. Why do we call a layer as nuclear layer? Where does the nucleus locate in neuron? It is inside the cyton. Okay. So it's inside the cyton where we have the nucleus. So where there is so many cytons, where there is a dense accumulation of cytons, we call that layer as the nuclear layer. So similarly, like the plexiform layer, we have 2 layers, outer nuclear and the inner nuclear layer. So the rods and cones have their nucleus in their cyton. We'll see about the structures in a couple of minutes. So they have their nucleus. Okay. That zone is called as the outer nuclear layer. The layer where there are accumulation or the density of nucleus of the rods and cones, we call it outer nuclear layer. And the inner nuclear layer is that zone where we have the nucleus or the cytons of bipolar cells. Okay, mainly bipolar cells. And we mentioned about the 2 other cells, 2 other neural cells, the horizontal and the amacrine cell. So this is where we are going to introduce that. So the horizontal cells, as we mentioned, they are between the rods cones layer and the bipolar layer. So this horizontal cell is also having its cyton in the inner nuclear layer. And the amacrine cell, it is between the bipolar cells and the ganglion cells. So that is also having the nucleus in the inner nuclear layer. Okay. So the nucleus of horizontal cell, bipolar cell, and amacrine cell together form the inner nuclear layer. So just pardon me, I've just changed the number of these 2 particular zones. Wait. Okay. The inner plexiform layer will lie below the inner nuclear layer. Okay. Will come here. Yeah. Okay. The inner nuclear layer is made of the nucleus or the cyton bodies of horizontal cell, bipolar cell, and amacrine cell. Right. So what is the role of this horizontal cell and amacrine cell? The horizontal cell is forming interaction with the plexus formed by the rods, cones, and the bipolar cell. Okay. So they do some transferring of impulses. They do some impulse interaction between the plexus formed between the rods, cones, and the bipolar cell. And in between the bipolar cell and the ganglion cell, there is interaction or impulse flowing. And between that, there is some cross interaction, and that cross interaction is by the amacrine cell. Okay, it's enough if you know this much. In detail, the functions will be dealt in the physiology. That will be in evening. Okay. Right. So this is about the layers. Okay. Now let's get back to this 10 layers. So if you want to write this 10 layers, first you should remember RBG Ha. Okay. So once you locate that in the diagram, then we will say the outermost layer is the outer pigmented layer. Within that we have rods and cones. Within that we have external limiting membrane. Okay, what is this external limiting membrane? Here is that layer, external limiting membrane. Okay, this zone, okay, that which I'm highlighting now is the external limiting membrane, and the innermost excuse me, this innermost lining is the inner limiting membrane. Okay, so we have now completed the external limiting membrane, the internal limiting membrane, and we also saw about all those plexiforms and the nuclear layers. So how do we remember? Outer pigmented layer, rods and cones, then we have external limiting membrane, then we have outer nuclear outer plexiform, inner nuclear inner plexiform, then ganglion cell layer, followed by that the nerve fibers, and the nerve fibers together they form the nerve fiber bundle that will form eventually the optic nerve. Okay, so that's about the 10 layers. Now let's see a histological diagram of this retina, right? So what you're seeing here, I took this picture from my record notebook. It's an eosin hematoxylin picture diagram of this retinal layer. Okay, so here in the retina, this is how a histological picture would look like. Okay, we have the pigment cell layer as the outermost aspect. Okay, this zone is the pigment cell layer. Okay, it is made of cuboidal epithelium. Okay, cuboidal epithelial lining is the lining epithelium of the pigment cell layer. Below that we have rods and cones. Okay, this zone. Then we have the external limiting membrane in between the rods and cones and the outer nuclear layer. Okay, outer nuclear layer. Then we have this plexiform layer. Nucleus, it's marked in hematoxylin, so it's more of those hematoxylin dots, and the inner plexiform layer. So the plexus is usually, it stains eosinophilic. So this is that zone, outer plexiform. Then we have similarly inner nuclear, and then inner plexiform, and within that we have ganglion cell layer, and down that we have the nerve fibers. So this is the zone of nerve fibers, and below that the lining is called as the inner limiting membrane. Excuse me. Okay, and now we'll see about okay, now we'll see the picture of a slide which has been stained with eosin hematoxylin, that which we can see under the microscope. Okay, you might see these in your lab. So this is the peripheral zone. Okay, it's not the foveal zone. We are showing the peripheral zones cross section, and we are seeing the H&E stained picture of that. Okay, all right. So we are moving on. That's about the histology, and we're now moving on to the rods and cones. Okay. So one question. In the first slide that we saw, I told you that cones are densely found in a particular zone in the retina. What is that zone? I mentioned some particular zones, right? Like the peripheral zone and other zones. So in which particular area the cones are found in abundance? Guys, are you able to see the screen? Are you able to see this diagram of the rods and cones? Yeah, very good. Johnson, SV, and Subramanian, Sarla, fine. Okay. So the fovea centralis is the particular area where we have the concentration or the density of cones. Okay. So it can, you can mention it as macula or fovea, it's all right. Okay. All right. So now what we are seeing is called as the rods and the cones. So we already saw it in the layers of retina. So here we are going to appreciate the structure of these rods and cones, and we'll see some basic things about this. We call this as rods because it is having a rod-like structure, okay? Right? And you can obviously see this pink colored one is the cone, and it is having this cone-like appearance, okay? The outer segment is having a cone-like appearance. So basically, they have three important parts. What are the three important parts? They have outer segment, okay? Based on the shape of outer segment, we call them as rods and cones. And we have an inner segment, and we have the synaptic end, okay? So this outer segment shape is what we have mentioned. Now, in the inner segment, what do we have? We have the cyton, or the part where the nucleus is present is in the inner segment. And we have the energy giving bodies, the mitochondria, and there is a connecting cilium existing between the outer and the inner segment, okay? So that is called as the connecting cilium, right? And on the bottommost aspect, we have synaptic vesicle. So the synaptic vesicle is what forms a plexus with cells lying within. So can you guys remember what cell we mentioned? That is rods and cones form a plexus with what cell? What nuclear cell or what um, special cell? I mentioned you RBG Ha. So of that, which cell is mainly interacting with the rods and cones? So the synaptic vesicle, the synaptic end is interacting with that and forms the outer plexiform layer. Can you guys remember? Okay, very good. Horizontal cell is one cell. What's another cell? The main cell? Yeah, bipolar cell. Yes. Okay. Yes, bipolar cell. So it's bipolar cell and the horizontal cell. Both of them interact with the rods and cones. Okay, that forms a plexus. Very good. Okay, now let's see some difference between the rods and cones. What is the difference, the basic difference that we can find between these two? Okay, first of all, rods and cones. We said cones are more densely found in the fovea or the macula. So that is a zone which is meant for the daylight vision or the bright light vision. So, and also we can call it as a color vision, okay? So cones are concentrated there, which means cones are meant for color vision. You can just correlate this way. Cones see, color see. So it's cones color vision. Okay, so cones is mainly meant for the color vision, and it's more densely found in the central part or the macula or the fovea. Okay, so that's about the location of cones. So what about rods? So cones are meant for color vision. So then opposite is for the rods. So rods are meant for the dark light vision or black and white vision. So rods are meant for dark, black and white, or dim light vision. And if cones are densely found in the center, then it's rods which are densely found in the periphery. Okay, all right. Now, because we mentioned the cones are for color vision, we know there are three primary colors: red, blue, and green. So based on that, we have three different types of cones. We have red cone, blue cone, and the green cone. Okay. So we have three types. But in case of rods, we just have one type. Okay. We have just one single rod. If it is stimulated, then you will see black color. If it is slightly stimulated, then you will see gray color. If it is not stimulated, you will see okay, if it's not stimulated, you will see black color. If it is stimulated, you will see white color, and if it's gradual stimulation, you will see some shades of gray. So rods is meant for seeing black or white or the shades of gray. Then cones are meant for the primary colors red, blue, and green, and based on those shades, we'll see other different colors. Okay. Now cones is occupying only a very little area in the retina, so definitely the count will be very less. It's 60 million. when compared to rods. What is the fundamental pigment in these rods and cones which is responsible for the particular color or the black and white that they are absorbing? So the cones have photopsin and rods have something called scotopsin or rhodopsin. Okay, so you just remember rhodopsin, rods. Okay, scoto means dark vision, so it's scotopsin, otherwise called as. Then cones, color, so it's photopsin. Okay, right. So all these functions of rods and cones will be dealt in the evening today. So now I hope you have got an orientation of the structures of rods and cones. Yeah, here is one quick question for you. Can you guys recognize this person? I hope most of you might be knowing him. Who is this person? Who is this guy or who is this man? Any guesses? We have a clinical correlation with him. That's why I'm asking. Yeah, yeah. So yeah, he's Mark Zuckerberg. Okay, the Facebook CEO, Mark Zuckerberg. Okay, so why is he important here for us? So yes, so blue in color and it's having this F written in white. So there is one reason for it. We'll see that. Just wait for a few minutes. First, we'll see something called color blindness. We have put a picture of Mark, the CEO of Facebook, because he is one of the victim of color blindness. Okay, so we otherwise call this color blindness as Daltonism. Okay, what is color blindness? We mentioned that there are three primary colors, right? So based on that, we have red cone, blue cone, and green cone. So color blindness is something associated only with the cones. Normally, we are all able to appreciate all the colors. So we are called as trichromats. Okay, trichromats, which means we can see all three colors. Trichromats. So it means or red cone, blue cone, green cone, all are normal. Then there are some people who are called as dichromats. Why dichromats? Because they can see only two colors. So which means of red, blue, green cone, one of the cones is not normal. It's not functioning properly. So based on that, we can divide dichromats into three group of people. Okay, one is red color blind people. That is, they cannot appreciate red color. Okay, or any shades of red color. Right, which means what is normal in them, in a red colorblind person, what are the two cones which are normal? What are the two cones which are normal in a red colorblind person? Then similarly, okay, any guesses for that? It's very simple. If someone is having red color blindness, there are two cones functioning normally. Red color blind means he is not able to see red color. So what would be normal in them? Yes, okay. Yeah, very good. So it's blue and green, Sarla and Harini, right? Okay, right. So it's blue and green. Both the cones are normal, so they are red color blind. In blue color blind person, red and green are normal. Blue is not functioning properly. Okay, fine. Right. There is another group of people called as monochromats. Monochromats means they can see only one color. So if they can see red color, then their blue cone and green cone are not functioning. They are not able to see green and red. That means they are blue color. I mean, they can appreciate blue color. So which means their red cone and the green cone are not good. Okay. So Mark Zuckerberg. Okay, I don't know the pronunciation property of his name. So Mark is having red-green color blindness. Okay, Mark is having red-green color blindness. So that is the reason why the Facebook logo is blue in color. Okay, because he can see only blue color, right? And you know, you can appreciate this in places where he goes, wherever he conducts a meeting, or most of the places wherever he organizes or arranges something, it will be blue. It will be the color theme will be blue, because he can appreciate only blue color. He cannot see green color or red color. Okay, common color blindness in the world. Okay, so here you need to carry this one thing. Red green color blindness is the most common color blindness. So you can correlate with this Facebook logo. Okay, and who is this person sitting there on the right corner, this black and white color photo? Most of you might not be knowing, because I knew this person only when I searched for his picture in Google. Okay, so he is Dalton. Okay, John Dalton. So if we say John Dalton, everybody will be knowing. John Dalton is a very famous physicist as well as a chemist, right? So he has done some work in biology as well. So we would have studied about him in school days, right? So this John Dalton is also having color blindness. So this Daltonism is named after him. Okay, so that is after diagnosing it in Dalton only. He was the first person having this. He took attempts to understand why he's not able to see certain colors. Then he gave a structure for he gave a reasoning for why this color is not seen. He thought that only his eye is taking only color blindness. So people thought if somebody is not able to see some color, they will not appreciate it, because somebody else might not know what I see, right? So only when something about the color comes to a discussion, people will know that a person is having color blindness. So this term of color blindness itself started only after Dalton's work. So it's named after him as Daltonism. Moreover, he has color blindness. So that's about rods, cones, and color blindness. Okay, now let's see a little about the blood supply. Okay, this is important because we mentioned two important layers, the outer pigmented and the inner neural layer, right? So the layers are supplied by two different arteries. One is by the central retinal artery, and the other is a ciliary artery. Okay, so how are these supplying? We can divide them into two zones. Okay, despite these layers, we said there are 10 layers. Despite these layers, we can divide this eye into two zones. Okay, so what are the two zones that we can divide? Right, we can divide them into the inner 2/3 and the outer 1/3 layer. Okay, so we are dividing them into inner 2/3 and the outer 1/3 layer. So inner 2/3 is supplied by the central retinal artery. Okay, so on the left side of your screen, what you're seeing is the fundus of the eye. Okay, so when you see through an ophthalmoscope, ophthalmoscope is an instrument through which you can observe the things inside the eyeball. Okay, so through the ophthalmoscope you'll be able to see the fundus of the eye. So in the fundus we can appreciate two important structures. So this is an actual eye picture. Okay, so here we can see the macula lutea. Okay, macula lutea with fovea centralis, this zone, and the one on the right side, slightly right side, we mentioned about 3 to 4 mm they are away from each other. So that's optic disc, right? So on the right side, okay, the picture that you're seeing is diagrammatic representation. Okay, so this zone is the optic disc, and what you're seeing here is the macula lutea with fovea centralis in the center, right? Okay, so you want me to zoom in? Can you see it clearly now? Harini? Arteries coming. Optic disc is important for two important things. One is the entry of retinal artery, central retinal artery, and the exit of the optic nerve, or the point where the optic nerve formation occurs. Okay, so these are two significant things in the optic disc. Okay, So here we are talking about the central retinal artery. So I'm showing it on the right side picture. So here, this point, optic disc, is where the retinal artery is coming. So as it comes, it's distributed this way. So first it divides into upper and lower, or the superior or the inferior branches. Then the superior branch will give the temporal branch. Okay, so we call it superior temporal branch, and another branch called superior nasal branch. So why is it superior nasal and superior temporal? Now I think most of you will be familiar with these terms. Temporal is this zone, okay, that I'm showing on my face. This is the temporal zone. Okay, you have the temporal bone here, and nasal aspect, that is medially, it is the nasal aspect. So as this central retinal artery of my eye comes out like this, okay, it pops out through the optic disc, and then what happens? The superior branch and the inferior branch are given. The superior branch will then divide into two branches. So medially, the one which goes medially is called the nasal branch. So the superior nasal branch comes towards the nasal aspect, so we call it that way, and the other one which goes laterally is called the superior temporal branch. Same happens on the inferior aspect. We have inferior nasal branch and inferior temporal branch. Naresh is asking medial and lateral, right? Okay. So optic disc macula lutea. So you can see the picture, right? On the medial aspect, we have the optic disc, okay? And on the lateral aspect, we have the macula lutea with fovea centralis, okay? Medial aspect, we have optic disc. That's a good question. You need to keep in mind. Medial aspect, we have the optic disc. So from the optic disc, the retinal artery comes. We have superior two branches, inferior two branches, right? So what the picture that you see here is the actual picture of how the retinal artery is coming, right? So I'm just highlighting that zone. So from here, it's going up and it's forming the superior. This zone, this branch is the superior temporal branch. And this is the superior nasal branch. Here inferiorly, the inferior nasal and the inferior temporal branch, right? So that's about the arteries. Now this is supplying, okay, this is supplying the inner 2/3 of the layers, right? Of the 10 layers, the inner 2/3 is supplied by the central retinal artery, that which we have talked about now. Now there's another supply for this retina. The outer 1/3 is supplied by this particular artery. Okay, you can see this in this picture. Okay, so look at here. There are something mentioned as posterior ciliary artery, right? Long posterior ciliary artery and the short posterior ciliary artery. So these are the arteries, ciliary arteries. They supply the outer 1/3 of the retinal layers. Okay, the outer 1/3 is by the ciliary arteries. Okay, that is by means of sinusoids. So what is a sinusoid? Sinusoid is nothing but a cavity which is filled with blood. So the capillaries then later form some sinusoids, and from the sinusoids, the nutrition is supplied to the retinal layers. So regarding blood supply, you need to remember just two things. The outer 1/3 is supplied by ciliary arteries, posterior ciliary artery, and the inner 2/3 is by the central retinal artery. Okay, now clinical correlation. So we have discussed all of them now. I mean, previously we have discussed that. So just a gist of all of them. Color blindness, what you need to remember is red-green color blindness is the most common color blindness. Okay, they are monochromats who can see only one color. You can relate it with the CEO of Facebook. Okay, so color blindness is associated with the cones. Then retinal detachment. What is retinal detachment? Detachment means some separation. So what separation? We said the 1 + 9 layers, right? So if the 9 layers get separated from the one layer, we call that as retinal detachment. Okay, so this retinal detachment is important because blood supply, as we mentioned, the outer 1/3, that means the outer aspect of the inner nuclear layer, that is of the 9 layers, some of the outer layers are supplied by the ciliary artery, and the ciliary artery is actually mainly supplying the outer pigmented layer. Okay, so in that case, if the retina gets detached, that is the outer and the inner layers are getting detached, some of the layers may be compromised from the nutrition. Okay, so for that reason, retinal detachment is clinically important. So you need to remember this. Retinal detachment is always the separation between the 1 and the 9 layers. Okay, if there is some separation between the inner layers. That is, in between the 9 layers, we don't call it as retinal detachment. Okay, we have a different term for it. For now, you just know retinal detachment. Then retinopathy. What is retinopathy? Pathy means some disease of the retina. So we saw the vessels are supplying the retina, right? So when there is diabetes or hypertension, or maybe in pregnancy we have some pregnancy-induced hypertension, these conditions are, they cause injury or damage to the peripheral vessels. So retinal vessels are also peripheral vessels. So like how they cause damage, okay? So that is affecting, that can compromise the nutrient supply to the retina. So that's also another clinical condition, right? So what's important for your exams? For your exams in anatomy, the most important thing is histology of retina. We showed a beautiful diagram for you. And by the moment, I would like to thank Janvi for creating that beautiful diagram. So it's from our illustration team. Okay. So that diagram is enough for your histology of retina, right? So you can draw the color diagram in your exam, and you can also practice the histological diagram with hematoxylin and eosin that I showed you. Okay. So those are important for your anatomy, but rest of the thing you need to keep in mind so that you will understand the physiology of this rods and cones, which will be dealt in evening today. Okay, right. So that's it for today, guys. Hope you all understood the class, and if you have any doubts you can ask now. Okay, fine. So if you have any doubts, if you get any other doubts regarding this topic, you can post in the comment box and we'll try to reply. And thank you so much. Have a good time. Have a good day.